I have coffee.
No,
Yeah.
anyway.
So now we can.
Welcome to Easy Immunology. My name is Stefan, and today we are looking in our first lecture at the innate immune system. And I'm not doing this alone. I have with me today Alex. Hi, Alex.
Hi, Stefan.
And Hannah. Hi, Hannah.
Hi.
They are both two fully trained biologists who will support me by trying to think of questions that you might have when listening to this lecture for the first or even the second time. And by this, help me to make this a bit more interactive and B, more interesting for you. And we're going to start right away with a question. Oh my God, these two have to answer questions. This is what my students always do not like to do. But I'd like to ask the two of them, what do you think is immunology or immune biology?
Immune biology or immunology is probably the studies of the immune system, most likely in the humans, but also maybe animals.
Mm-hmm. That is quite correct. So the official or one of the official definitions is the study of all aspects of the immune system, including its structure and function. This is what Hannah meant. And what is also coming on top is that it also includes the disorders of the immune system. So there is, as we will learn in future lectures, there's a whole slew of diseases that are caused by the immune system either over or under functioning. And this is a fascinating topic. So what does this mean? So if we have, like here, contact with a pathogen like a bacterium or a virus, we want to understand as immunologists or immunobiologists, we want to understand what's going on, how our immune system deals with these pathogens, what kind of reactions are going on, what kind of cells are involved, what kind of molecules do we need to fight these pathogens. And this is an example here that you will understand after maybe this lecture already, or at least after the second or the third lecture. And if we understand this, we can use this knowledge to develop vaccines. So we can simulate non-dangerous types of infections with these kinds of vaccines that will train our immune system to mount a so-called memory response that will protect us from future encounters with said pathogen. And this is very important. This is one of the most influential, most important discoveries of the last century and has saved millions or even billions of lives in the last years. So we will start with the origins of immunology, and the father, if you want to say it like that, of immunology is Edward Jenner. Here we have a picture of him, and he was a British doctor, and at that time the smallpox were a big problem in society, also in the British society. What is smallpox? Smallpox is a disease that is caused by infection with smallpox virus. And this results in severe disease, and the patients are characterized by having these pox on their skin, and this is all over their body, and this is a severe infection, and people often die from it. And even if you don't die from it, you do not have a good time for a few weeks. So this is a severe problem. And he wanted to improve this. And what did people do to treat smallpox at that time? They did something that is called variolation. And what is this? They took like one of these pox and carefully opened it and dried the fluid that was in these pox, and then gave this dried, it's basically virus, preparation to the patients by either inhaling it via the nostrils or transferring it to superficial skin wounds. And this also leads to the smallpox in a way, because this is like a controlled infection, and you can do things like somehow treating the content of these pox to make it less infectious. You can use pox from people that did not have such a severe disease. You can do different things, but in the end, this is just an infection, and in most cases, this also leads to smallpox infection, which leads to 1 to 2% of people dying from this variolation. And this is also not optimal, as you can imagine. But this was all that was available at that time. What Edward Jenner observed is that milkmaids, these were persons that draw milk from cows all day long, this was a profession at that time, they often get infected with cowpox. This is a related but different virus from the same family that can cause disease in these milkmaids, but the disease is less severe. You can see there are less amount of red dots. The disease is not so severe, and you can usually easily shrug this off after several weeks. And what he observed is that if these milkmaids then get in contact with the smallpox, they do not get infected anymore. So you can see here, there are no red spots, no infection. So this means that by having contact with the smallpox first, the immune system of these milkmaids learned something, and that helped them to protect them from smallpox afterwards. And he made this very important discovery. He published a paper on it. People didn't believe him in the first place. He did more experiments where he infected persons under controlled conditions, first with the cowpox and then with the smallpox. In the end, he could prove his theory, and this led to the first vaccination against cowpox. And this is also where the word vaccination comes from. It comes from the Latin term vacca, which means cow, as in cowpox. And this is a very important discovery that led to a worldwide eradication campaign against smallpox. And one of the biggest successes in modern medicine is that in the year 1979, the WHO, the World Health Organization, could declare the smallpox as eradicated. Vaccination against smallpox was the first one, but many followed. I have brought here two more examples. One is a vaccine against diphtheria. This is an infection of the upper airways of the throat that can lead to severe disease and even death in some cases. And you can see that there are many cases until the mid-1940s, where the first vaccine was introduced. And you can see that the number of infections severely declines afterwards to a level that is barely not measurable. And the same is true for polio. You all know polio. This is a kind of paralysis caused by the polio virus. And there were two vaccines. First was an inactivated vaccine. So these were polio viruses that were treated to be not infectious anymore, but had some side effects. And then later on, there was a second generation oral vaccine that you can just swallow. And you can see already after the first vaccine, the number of cases drastically dropped to nearly zero. And this is important because whereas diphtheria, you might not have long-term complications from polio, you usually have them. These are the people that lived in iron lungs for their natural lives. And this is, of course, a very bad thing. And this vaccine was able to prevent this. That immunology is important is not something only you have realized by listening to my lecture, but it's also something that the people that give the Nobel Prizes have realized. So here we accumulated the number of Nobel Prizes that were given for discoveries that are somehow related to immunological discoveries. And you can see that already one of the first Nobel Prizes to Emil von Behring was given for the diphtheria vaccine. And then we have Paul Ehrlich and Ilya Mechnikov for the discovery of the principles of immunology. We have Karl Landsteiner for the blood groups. We have the people discovering the first antibiotics. We have people working on immunological tolerance, on antibody structure, on the method to generate monoclonal antibodies, which are important molecules in human therapy at the moment. The discovery of the HI virus, and recently the discovery of the dendritic cells and innate immunity. This is also what we want to talk about today. So there is an increasing knowledge, and people realize how important it actually is for human health. So to start our discussion about the innate immune system and the immune system in general, we have to look at what the problem is. Why do we need an immune system? And the problem is that we are constantly exposed to a whole slew of microorganisms that want to live in our body, on our using our resources, and some are making us sick in the progress. And here I would like to ask the two of them again if they know different types of pathogens. This is, of course, something they know very well. Yes, Alex?
I would say bacteria, viruses, and worms.
Mm-hmm. bacteria, viruses, and worms. What else do we have?
Prions?
And one thing is missing, and one thing could be further differentiated.
Like
multicellular and
Yeah, exactly. Just do it.
multicellular and single cellular organisms.
Yeah, this is what we call parasites. There we have multicellular and single cellular parasites. Yes, and the last thing we are missing is
fungi.
Fungi, yeah.
True.
Fungi.
So here I've put them, and you can already see from my crude representation that they are very, very different in their structure and how they look like. And if we just take one characteristic of these pathogens, their size, and group them by their size, you can see that this is very, very different for all of them. So prions are some few misfolded proteins. Viruses are some 100 proteins that fold into a complex structure. Then we have bacteria, which are real single-celled organisms. They do not have a nucleus, but they are fully functioning single-cell organisms. Then we have the single-celled parasites. They are much, much more complex. They are real eukaryotes. They have a nucleus. They have many, many more genes than a bacterium has. They are also quite a bit bigger. They are the size of our cells in our body. Then we have fungi. They can also be single-celled. This is what we call a yeast, but they can also be multicellular. Here we say a multicellular fungi, and they can even be really big, forming these micelle structures that can be hundreds of meters in size. And then we have the multicellular parasites. So they start at a few millimeters. If you have some gut worm or some tapeworms, and they can be even bigger than humans are. So they are big, big tapeworms that can be up to 12 meters. And you can see that this is a big problem for the immune system, because these pathogens are so different, and the immune system has to have different tailored mechanisms to fight these pathogens. And this is what we will look at over this lecture and also the next. So then I would like to ask again the question, what do you think is the function of the immune system? Yes, Alex?
To fight these pathogens and to keep the body healthy.
Okay. And in order to do so, the immune system has to do one more thing.
Know what is tolerance.
Yeah, or know what is self and what is not self. So in order to be able to fight these pathogens, you have to distinguish what is normal, what is my own cells and tissues, and what is not normal, what is foreign, what is non-self. That's exactly true. So this is something, of course, Alex knows after doing more than 100 videos with me. So the function of the immune system is to protect us, which we call the host, from foreign organisms by a variety of different effector cells and molecules, and to distinguish between self, that is all the structures of the host, all our cells, all our tissues are self and need to be tolerated, and non-self. So this is basically everything else. And there, the immune system even has to distinguish between non-self components which are not dangerous. So this is your breakfast or your lunch, or non-self components which are dangerous. These are pathogens, for example. So this brings us to the multiple roles of the immune system in health and disease. So in health, it has the function to protect us from infection and the development of cancer. And here we will look at today at the innate immune system. Then we will continue in the next lecture with T cells, immunological tolerance, and NK cells. Then we'll look at the B cells. And finally, we will close with a new concept in immunology, which is called trained immunity. These are very recent results that have now made it into a coherent theory that allows us to explain why even the innate immune system can form something like memory responses. After understanding how the immune system protects us from all these pathogens and cancer cells, we will switch to the pathological sides and try to understand how an overreaction of the immune system can lead to allergies if it reacts to non-self components, or autoimmune diseases if it reacts to self components. We will look at the interaction between tumor cells and the immune system and how tumors manage to evade our very efficient immune system. We will look at the immune system as a barrier to transplantation between individuals within one species or even between different species. And we will finally have a look at diseases that arise from one or several components of the immune system either not functioning correctly or even completely missing. This is what we call immunodeficiencies, and these are very, very severe diseases usually for the affected patients. So if we start now our journey into immunology, we have to define one word, and this word is antigens. I think so far I've managed to not say the word antigen, but now I cannot promise any longer. So what is an antigen? It is historically a substance that you inject into either humans or animals, and this results in the production of antibodies. So historically it is defined as an antibody generating substance, and these are usually non-self components because, as we will learn in the third lecture, all our own cells and tissues in our body is what we call self, as we just learned. They are tolerated by the immune system because our immune system learns what is self and what is non-self. And therefore, these self components usually do not lead to the production of antibodies. One exemption are alloantigens. So these are antigens that are different between members of one species. So this leads to me looking different than Alex or having a different hair color than Hannah. And these are specific for that certain individuum, and therefore they are only tolerated in this individuum, and therefore they can result in antibody responses or immune responses in general. And this will lead to, for example, transplant rejection between non-identical individuals. So antigens can be any substance that is recognized by T and B cells. In many cases, as I said, these are foreign proteins. They can be derived from pathogens like viruses, bacteria, or parasites, but they can also be derived from environmental proteins that are actually not dangerous, like pollen, plant food, or animal dander. And this is then what we call allergens. And this is a big problem because allergies are constantly on the rise, and nearly every second one of us is allergic to something meanwhile. So our immune system consists of different components, and I have defined 4 of them that we want to look at today. At least the first 3 we want to look at today. The first one are so-called anatomical barriers. These are our skin, the mucosa in our mouth, the respiratory and the intestinal epithelium, and these have the function of just shutting the pathogens and everything that is not belonging in the inside in your body out. So they are barriers that prevent these pathogens from just entering your body. Then we have the complement system and antimicrobial proteins that are the first or 1.5 line of defense against pathogens, and they eliminate many pathogens before they can even get much further into your body. Then we have the innate immune system, which consists of different cells like dendritic cells and macrophages that we will look at today. And then we have the adaptive immune system, which consists of T cells, B cells, and the antibodies that are produced by B cells. And as I just said, today we will look at the first 3 barriers or first 3 components, and then we will have understood a lot about the immune system already. And the last one, the adaptive immune system, is, as I have to say, unfortunately much more complicated. And whereas I can do the first 3 barriers in one lecture, I will need nearly 3 more lectures for the last barrier, but we will manage. So let's start with the first barrier. This is formed mainly by epithelial cells, and epithelial cells are these blue guys, and they are fascinating because they are a layer of cells that is just one cell thick, and they form the layer, the protection, or the frontier between the sterile inside of your body and the non-sterile outside of your body, and they just prevent pathogens from coming in. And they do so by holding very tightly together, and this is achieved by proteins that we call tight junction proteins. And here I've put in a microscopical picture of some epithelial cells, and you can see in gray is the nucleus, in red is some cytoplasmic component, so something that is inside the cell, and in green we have these tight junction proteins, and you can see that they are sealing these cells, or the very small rooms between these cells, from letting through any pathogens. So these cells stick together very, very tightly and do not usually allow any protein, any pathogen, or anything else to come into the inside, the sterile inside of your body. The epithelial cells are just one type of barrier in the innate immune system, and we distinguished three types of barriers. And the first one are mechanical barriers, and these are the epithelial cells, for example. And we find these barriers on all places in our body that have potentially contact with the outside. So we have the skin. Of course, this is in contact with the outside. We have the gut, which is in contact with the sterile food or the non-sterile food we consume all the time. We have the lungs, which take in the non-sterile air we breathe. And we have the eyes and the nose, or even the sexual tract, the reproductive tract, that is in contact with the outside. Besides the epithelial cells, we have on the skin and the gut, we have the longitudinal flow of air and fluids. So if I give my lecture, I'm dancing around here. This generates some flow of air that will just shake off some pathogens. And this also happens in your gut. By moving the gut with your muscles, then also pathogens will just be cleared away before they can attach to your gut cells. Then in the lungs, we have mucus that is produced. Mucus is like a fluid that is used to trap the pathogens and to clear them away. And this mucus is moved by cilia. These are like hair-like structures, small hair-like structures, that push the mucus out of your lungs into your upper airways, and then you can cough them away. If you have like a respiratory tract infection, you notice that you can produce a lot of mucus in a very short amount of time, and this is just to get rid of these pathogens. In the eye and the nose, we have the tears. You also notice, if it's not only if you're sad, but also if you have something in your eye will start tearing, and this is a mechanism to get rid of many pathogens before they can colonize your ear or your nose. And also in the nose, we have some cilia that just transport pathogens away. The second type of barriers are chemical barriers. So they consist of chemical molecules that either directly attack pathogens or interfere with their optimal growth. So in the skin, we have, for example, fatty acids. These are molecules that integrate into the membrane of bacteria, and this disrupts their normal membrane structure, preventing their growth, their division, their proliferation, something like this. Then in the gut, we have a low pH, so it's an acidic environment that prevents many bacteria, not all of them. Some have adapted to this niche to grow there. Then we have enzymes like pepsin that degrade a lot of proteins. So in the lungs, we have the pulmonary surfactant. So this is a mixture of lipophilic and hydrophilic molecules that integrate in, for example, the mucus. And this reduces the surface tension of this mucus layer, and this prevents bacteria and other pathogens from sitting on the mucus. They will just sink in, and then they can directly be eaten by immune cells. So this is a fascinating topic. In the eyes and the nose, we have enzymes in the tears. The most famous one is lysozyme. You can also buy it in the form of tablets that you can give to small children, and these will directly lyse certain bacteria and therefore get rid of them. And in all places, we have antimicrobial peptides, which we will look at in more detail in a few slides and try to understand what they do, but basically they destroy pathogens. The third type of barrier is the microbiological barrier. And what you have to know to understand this is that all these points of contact with the non-sterile outside, the skin, the gut, the lungs, the eye, the nose, they are not sterile. They are colonized by microorganisms, bacteria, maybe even some fungi, some virus. We don't know about viruses, but I assume there are some. And these just colonize this niche, and they prevent, by just being there and having taken the space, they prevent the bad guys, if you want to call it, pathogens like this, from colonizing your skin, your gut, or something like this. So this is very important. And if you take, for example, a lot of antibiotics, you destroy your microbiological barrier, and this can cause the problems, because then you open this niche to guys that are maybe not so nice to you and your body. So besides the anatomical barriers, we have the complement system and the antimicrobial proteins, and they act once a pathogen has managed to cross these anatomical barriers. So the fight directly starts for these pathogens once they're inside our body, and we will start with the complement system. And I know that this is difficult for a lot of students, and we will try to make it as simple as possible. But as you can imagine, and as is true for everything I'm going to tell you, it is infinitely more complex than what we are telling you here. So you can spend, I've done this with my students, you can spend 3 hours talking about the complement system. This is like 60 pages in the most famous immunological textbook, and you will come out more confused than as you went in. So it is very complicated. But the complement system can directly inactivate pathogens, and there are 3 ways how the complement system can be activated. And first we are talking about the classical pathway. And it starts on the surface of the pathogen that has invaded our body. And it starts with antibodies binding to some proteins of that pathogen. You can see here the surface of the pathogen with its lipid membrane. Then we have these 2 proteins that are specific to this pathogen, and for some reason that we will understand in 2 more or 3 more lectures, we already have contact with a similar or the same pathogen, and we have made antibodies against this structure. And these antibodies are molecules that specifically bind to the proteins of that pathogen, and they trigger now the activation of the complement cascade. And this results in the fixation of these complement proteins on the surface of said pathogen. So you basically add a tag to the pathogen that says, oh, this is strange, this is dangerous, this is foreign, it should be eliminated. And there is a very complex sequence of how these proteins are added and how they are modified. And the complex or the complement proteins are abbreviated with a big C, and then they have numbers. And the first proteins that are attached to the surface of the pathogen are C2A and C4B, and together they form the so-called C3 convertase. This is called C3 convertase because it takes another complement protein, C3, one of the most important complement proteins, and it cleaves it, it converts it into a big fragment, which is called C3b, and a small fragment, which is called C3a. The C3b then sticks to the C3 convertase, consisting of C2a and C4b, and forms the C5 convertase. And this does exactly the same thing again. It takes a next complement protein, C5, and cleaves it into a big molecule, which is called C5b, and a small molecule, which is called C5a. And this then continues by recruiting and cleaving and building together like molecules like C6, C7, and C8. And together, this C5b, C6, C7, and C8 now integrate a molecule into the membrane of the pathogen, which is called C9. And this C9 is a pore building protein. So many C9 proteins that aggregate form a pore into the membrane of this pathogen, and this will lead to, these two know it because I tell it like in every lecture, of the inner components of the bacteria flowing out uncontrolledly, and this usually results in the death of the pathogen, and this is a process that we call cytolysis, so the lysis of the cell by this C9 protein, and this combination of C5b, C6, C7, C8, and C9 is what we call the membrane attack complex, because the membrane of the pathogen gets disrupted. And Alex has a question.
Does this complex only work for bacteria, or are there other pathogens which could be attacked?
Yeah, it also works to a certain degree for fungi and maybe also for parasites. But you have to imagine that here we have a bacterium, which is a rather primitive organism. It doesn't have so many genes, but a fungi and a parasites, they are real eukaryotes. They have like 100 times more genes, and they have developed mechanisms to directly get rid of these complement proteins again. So therefore it works to a certain extent, but it doesn't work as good as for bacteria, because they just have more mechanisms to get rid of these complement proteins.
Thank you.
Okay, so this is highly complex. As I said, it's more complex than we have time and energy to tell you today. But what people refer to with this is the so-called complement cascade. And the word cascade refers to the fact that we start with very few molecules of C2A and C4b, and they cleave more molecules of C3. Then we have more C5 convertases, they cleave even more molecules of C5. And this is something that perpetuates itself with every cycle. So we start with very few molecules and we cascade down, and every step is more and more and more. So we end with a lot of C9 molecules that are integrated into the membrane of the pathogen. That's why people call it a cascade. So the complement activation has three main purposes. The first one is opsonization. Opsonization is a word that in immunology refers to marking some structure, some pathogen as foreign and dangerous. So by turning on the complement system, we have now these complement proteins on the surface of the pathogen, and they were not there before, and they just tell other cells of the immune system, hey, look, this has complement on its surface, it's probably something dangerous that should be eliminated. The second one is, what I didn't tell you in the slide before, is these small molecules that result from the cleavage of C3 and C5, the C3A and C5A. They diffuse away from the pathogen. So the pathogens are not only finding themselves with complement proteins on their surface, but they find themselves in a cloud of C3A and C5A molecules, and this cloud is very dense in their near surrounding, and the further and further we go away from the pathogen, there's less of these molecules. And the other cells of the immune system have receptors for these molecules, and they travel towards the gradient of higher concentration, and by this way they find the pathogens, and they then dock to the pathogens, and then they can start eating these bacteria or killing them with the mechanisms we are going to learn in a few slides. So this is how the complement system can get rid of many pathogens. And I made the inflammation and tissue injury part in the middle. I made this red because if the complement system is not activated correctly or by the wrong stimuli, this can cause a lot of different diseases in humans, and therefore it's a problem. For example, red blood cells do not have a nucleus, and they are usually the first ones that get eliminated if the complement system is not correctly controlled, and then these patients have different kinds of anemias. Besides the classical pathways, so where antibodies trigger the activation of the complement system, we have two more pathways. The first one is the alternative pathway. And if you look very closely, the uppermost picture and the one in the middle, you see that the only difference there is the lack of the antibody that triggers the activation or the fixation of the C3 molecule. And you might ask yourself, now how does this work? This works because the C3 molecule is inherently unstable in a aqueous solution. So it just by itself dissociates into C3A and C3b, and this can happen by just being in, or when being in proximity to the surface of a pathogen. And this will just lead by chance to the fixation of C3b on the surface of the pathogen, and this will allow the complement cascade to start. It's not as efficient as when it is started with antibodies, but it also works. The question is now, why doesn't the complement system then kill our own cells? Hannah already raised her hand. The thing is, our own cells in our body have developed mechanisms to just remove the C3b directly again. And this is something that bacteria do not have. And the third pathway is the so-called lectin pathway. And you can see by comparing the first and the last picture that here the antibody is replaced by a lectin, a mannose binding lectin. This is a structure that has specialized in recognizing sugar structures, sugar-like structures that are only present on these pathogens. And then again, it starts the whole complement cascade. Besides the complement system, we have antimicrobial peptides that can directly kill different pathogens. And these are fascinating little molecules that have a very polar structure. So they have one part that is highly positively charged and one part that is not so positively charged. And they can integrate into the membrane of pathogens. Here the red lipid membrane is from a red bacterium, and if 2 or more of these molecules integrate, then this leads to these molecules with their positive charges repelling each other, and by this they open up the lipid membrane of that pathogen, and by this they are generating holes where again the inner components of the bacterium in this case flow out, and this is of course not so healthy for the bacterium. This is one mechanism that is discussed. There are more that are more complicated, and at the moment it's not completely clear. And I think here there is a lot of potential in future for new antimicrobial molecules, and to better understand this would be highly beneficial. Okay, this is already the second component, and now I would like to spend the rest of the lecture with the innate immune system. And as you have already seen, from one barrier to another, it gets more complicated. And now it gets even more complicated because now we are looking at a slew of different cells that are very complex and have evolved different mechanisms of dealing with pathogens. So most of the cells of the immune system, doesn't matter if innate or adaptive, can be found in the blood. And therefore we will first look at the composition of blood. It contains or consists of plasma, 55%. So these are all the water-soluble parts. And we have the cellular parts, which is around 45%, which are all the cells in the blood. And what do we have in the plasma? We have water, this is the majority. We have electrolytes that we need for functioning of many cellular processes. We have nutrients like fatty acids, amino acids, sugars. We have metabolites, so some molecule that was generated by the metabolism of a certain organ or a cell that get transported from one place to another in our body where they are used. And then we have plasma proteins. So there are, for example, albumins. These are the most frequent proteins in human blood that usually also get measured in a small blood count. They are important for keeping the osmotic pressure in the blood, for transporting certain proteins, certain molecules. Then we have antibodies. This is around 10% of all proteins in the blood, important for the recognition and fighting of pathogens. Then we have fibrinogen regulatory factors and clotting factors, which are used for blood clotting, so closing of small or big wounds need all these proteins, and this is a complex cascade again. And these are the plasma components. And in terms of cells, we have, as you know, probably the red blood cells, which transport oxygen. We have the leukocytes, so the white blood cells, that are the immune cells, as we will learn in a minute. And we have the platelets, which are small guys that are used to as material to close wounds. So besides the components we just talked about, the immune system mainly consists of different cells. And in the innate immune system, we have at least 8 different cells. There are more, I know. Try to make it a little bit simpler than it actually is. But we have, for example, mast cells, basophils, eosinophils, neutrophils, macrophages, dendritic cells, NK cells, and NKT cells that are part of the innate immune system. And then we have the adaptive immune system, which consists of the T cells and the B cells. And the T cells, as we will learn in the next lecture, can be differentiated again into different subtypes with different functions. And the B cells are the producers of the antibodies. And historically, because people didn't understand where antibodies come from in the first place, antibodies, or the B cells and the antibodies, have been summarized under the humoral immune system. Humoral means in the humerus, so in the body fluids. That's where most of the antibodies are found. And the T cells have been summarized as cellular immunity, but we know now this is not completely true, because also B cells are of course cells. But this is just historically from not being able to closely characterize and closely differentiate all the involved cells like 100 years ago. So as we have just had it in 2 slides back, all the cells of the immune systems are summarized under the term leukocytes, so white blood cells. And immunologists like to use fancy words. I think I've managed to avoid the term leukocytes and granulocytes and stuff until now. But I would like to introduce them to you so that I can start to use them. So besides the leukocytes, so all the cells of the immune system, we have granulocytes. These are cells that have like small granula in their insides. These are neutrophils, eosinophils, basophils, and mast cells. Then we have phagocytes. So these are cells that are able to phagocytose, so eat up pathogens. And these are dendritic cells, macrophages, and neutrophils. And then we have the lymphocytes. So these are the cells of the adaptive immune system, the T cells, the B cells, plus the NK cells, which are somehow between the innate and the adaptive immune system. So for the moment, it's okay to understand that the NK cells are some like evolutionary progenitor cells on the way to T cells that are still in our body present at the moment, and they have the function to fight different pathogens and tumor cells. Yes, Alex?
Can you tell what is in the granula?
Yeah, that's different. So granula are like small sacs, if you want to call it like this, inside the cell. And they contain usually a mixture of molecules that are used to either perform processes within the cells. So we have, for example, lysosomes, which contain proteases, so proteins that degrade other proteins that are used to degrade the pathogens once they're inside the cell, for example, in terms of the phagocytes. And then we have granula that are released to the outside of the cell, and they contain a lot of molecules that are used to just damage or disrupt pathogen function. So they are molecules, as we will learn with the neutrophils in a few slides, that digest certain proteins. There are molecules that generate reactive oxygen species. So it's a toxic cocktail, if you want to call it like this, that is used to then fight pathogens. Yes, Hannah?
My question is, you also have these small little bubbles drawn in the macrophages, but they do not belong to the granulocytes, whereas
Yes.
the neutrophils do.
Yes. It's complicated, and it's like also historically, but the thing is, as we will see in a few slides, the macrophages have the function to take stuff in and to just do something with the pathogens inside the cell that we will understand in a few slides. And this just leads to the pathogen being gone. It just gets eaten up. The neutrophils do the same thing, but the neutrophils also give stuff to their surrounding and thereby kill pathogen, because you can imagine you can only eat so much. Yeah, if you have 1.5 or 2 pizzas, you will not be able to eat more. But the neutrophil can just keep on producing these molecules and shed them in its surrounding, and therefore kill a lot more of these cells. So that's just the difference. Yeah. Okay. Here I have made you a small graph with the frequency of immune cells that we can find in human peripheral blood. Peripheral blood means you just take some blood from my arm. This is the peripheral blood, and you just then, with certain techniques, identify and quantify the different cells, and you can see that the neutrophils are by far the most frequent cells in human blood, followed by T and B cells, some NK cells, some monocytes, and the rest is not so much. Yeah, this might look different in different organs. This might look different in different species. So if you look at samples from a mouse or from a cat or from a dog, it might look different. It will look different because, for example, in the mouse, the T and the B cells are the most frequent cells in the peripheral blood. So this is highly species specific, and it's also dependent on the health status of this individuum. If you have For example, somebody that is highly allergic, he or she will have an increased number of eosinophils, not to the extent that neutrophils are here, but this is something that can be recognized, that can be quantified, and this is also done in clinical practice. But this is just an example to give you an impression how the numbers actually are. So we will then look at the different cells, and we will start with the phagocytes. As I said, these are the dendritic cells, the macrophages and the neutrophils. And we can distinguish them into two groups. So phagocytes are cells that eat up stuff. And the first group is doing it to present antigens. What this means, we will learn in a minute. And the other one is doing it as an effector function. So what is the difference? The first one is just taking up pathogens to do something with them, to induce an immune response, to show it to other immune cells, basically. And the second one is just eating up as much as they can so that the pathogens are gone. So this is what we would define as effector function. So the first ones that do antigen presentations are the macrophages and the dendritic cells. And they arise from precursor cells that we call monocytes. And the function of all of these cells is to take up a lot of stuff in their surrounding. And once a monocyte recognizes something dangerous, something different, something non-self, it differentiates into a macrophage or dendritic cell, depending on where it is, what stimuli it finds, what stimuli it also gets from other cells. And then they present what they have found to other immune cells. And there are different types of dendritic cells and macrophages. And just to name you some example, it depends on the tissue. They are slightly different, or even not so slightly different in different tissues. For example, in liver, they are called Kupfer cells. The second F is correct. Yeah, this is after the scientist that found them in the liver. That was a Dr. Kupfer. In the lung, they are called alveolar macrophages. In the central nervous system, so in the brain, they are called microglia cells. They are some like Frankenstein cells between macrophages and dendritic cells. In the bone, they are called osteoclasts. So these are macrophages that are specialized on degrading, for example, bone tissue, which is important if you want to, for example, grow. And then your bones have to grow. Maybe you have to get rid of some bone tissue in this process. So there are different cells with specialized functions in the different tissues. So how do these cells now decide what is dangerous and what is not dangerous? And they do this by using so-called pattern recognition receptors, PRRs. Here we have one integrated into the membrane of one of these cells. And what these pattern recognition receptors do, they recognize pathogen-associated molecular patterns called PAMPs. So these are structures that only exist in certain pathogens, but do not exist in our own cells and tissues. And I've brought my favorite example. It's flagellin. It is the protein that forms the bacterial flagella. This is a whip-like structure that these bacteria use to move in their surrounding. And this whip consists of 100,000 molecules of flagellin. It's always the same molecule that forms like a spiral. And this is only existing in bacteria. In our own body, we also have whip-like structures, cilia, that we just talked about in the beginning of the lecture, but they are totally different. They are these classical 9 × 2 + 2 microtubuli structures, very different structure, much more complicated. Therefore, once an immune cell recognizes a flagellin molecule or another PAMP, it knows, okay, there's a bacterium around and I need to form an immune response. These PAMPs then bind to these pattern recognition receptors and initiate the activation of the immune cell, and then it can start fighting this pathogen. Here, Alex has a question.
You called about PRRs, but beside the receptor that is binding flagellin, there is TLR. So
Yeah.
Can you talk about TLRs?
Yeah, I wanted to do it in the next slide. So basically, as a teaser, TLRs is an abbreviation for toll-like receptor, and this is the most famous or the most well-researched group of these pattern recognition receptors. And here you have, for example, dendritic cells and macrophages, and you can see that it is not only one type of pattern recognition receptor, but many different types, and they can be on the surface of the cell, and they can also be inside the cell, and they can look quite different from each other. And here we have some examples. So for example, we have the toll-like receptor 5 that binds flagellin, but we also have toll-like receptor 4 that binds lipopolysaccharide. We have glucan receptors that binds bacterial glucan structures. We have mannose receptor, which recognizes mannose-related proteins, and there are many, many molecules. And to structure them a bit more, we have different classes. And the first one are the so-called toll-like receptors. And as I said, these are the most well-known, the most well-researched class of pattern recognition receptors. And they're called toll-like receptors because they were discovered in a lab where the boss, that was Professor Nusslein Vollhardt. She's also a Nobel Prize winner for her discoveries in Drosophila biology. She was called by one of the postdocs, and what they did a lot of mutants of Drosophila, and in one of the mutants they generated, they destroyed, for example, by radiation, they destroyed the toll-like receptor analog that Drosophila has, and then the immune system of that fly doesn't work, and they cannot protect themselves from fungi anymore. And you see this under the microscope if you look at these flies that are covered in fungi because they cannot fight them anymore. And when the postdoc showed this to Professor Nusslein Vollhardt, she exhaled and said, "Wow, this is toll." So toll is the German word for great, and that's why they are called toll receptors in Drosophila, and the analogs in humans and also other animals, higher vertebrates, are called toll-like receptors because they have a very similar structure. And these toll-like receptors can be further subdivided into two groups. The first one are toll-like receptors that are on the outside of the cell, and they recognize and interact with structures that are also on the outside of the cell. So what is outside must bind to something that is outside. And then we have toll-like receptors that are inside the cell, usually in the endosome. This is a vesicle-like structure in the inside of the cell where stuff ends up that the cell takes up from the outside, and also a lot of viruses replicate there, and stuff that is taken up gets recognized there. And these usually are specialized in recognizing DNA and RNA structures. For example, double-stranded RNA is something that does not occur in our replication cycle, but it does occur in the replication cycle of certain viruses, and therefore it can be recognized. Or CpG DNA, this is cytosine phosphate guanine. This is a motif in DNA. It also is present in our cells, but here it's usually methylated, and bacteria have not developed this methylation mechanism, and therefore this non-methylated CpG DNA inside an endosome is usually a signal for being infected by, for example, a bacterium. Besides the toll-like receptors on the surface and inside the cell, we have some more pattern recognition receptors. So we have C-type lectin receptors. Hannah worked with them in her master thesis. They are specialized in recognizing different polysaccharides, so sugar-like structures, outside of the cell or on the surface of target cells. Then we have the knot-like receptors, or the NLRs. They form something that we call inflammasomes that we will look at in a few minutes. And they are recognizing a lot of different structures that only are inside the cell once the cell is infected. So certain pathogens, certain bacteria, such as certain parasites, try to hide in our immune cells by infecting them. And this can be recognized by these knot-like receptors. Then we have the Rig-like receptor family, the RLRs. They are a growing family of receptors that recognize different types of viral RNA. And we have the cytosolic DNA sensor, which recognizes certain microbial DNA motifs. They are, for example, very important for recognizing Ebola virus. So this is the types of receptors that the innate immune system uses to recognize pathogens as foreign and dangerous. And what one has to say is that these receptors are the same in all of us. So I have the same set of toll-like receptors and C-type lectin receptors as Hannah has and also as Alex has. So if not one of us has accumulated a mutation, which can happen, we should all have the same receptors to do a first analysis of most molecules and pathogens that we get in contact with. So what happens now if an antigen-presenting cell, so a dendritic cell or macrophages, comes into contact with a pathogen? Here again, the bacterium with the flagellum that triggers the activation of our pathogen recognition receptor. It's toll-like receptor 5, as we know, and this triggers different things. The first one is it transmits an activating signal into the nucleus. This is highly complex, something that we also look in our own research, but do not have time to go into detail here. But it results in the activation of many, many different genes. So if you have a virus-infected cell, of the 20,000 or something genes we have, unto 5, 6, 7, 8,000 can be activated of them. So this is a drastic change in gene transcription that happens once they recognize a pathogen. This, among other things, leads to the expression of so-called co-stimulatory molecules on the surface of the cell. And this is just, if you want to put it easy, it is a translation of the sorting process of this antigen-presenting cell, so recognizing the pathogen, deciding if it is dangerous or not. And if the cell decides, okay, the combination of the pathogen recognition receptors activated by that bacterium allows me to conclude that it is dangerous, I will just translate this complex information of what it is and how dangerous it is into a single molecule, a co-stimulatory molecule that I show on my surface, and this just says, hey, I have found something that is dangerous. That is what a co-stimulatory molecule does. Then also molecules that are produced are cytokines. These are small, highly soluble molecules that transmit information between cells in the immune system and also non-immune cells. And these cells also constantly take up all the stuff they find in their surrounding, including pathogens or parts of the pathogen. Then they get degraded inside the pathogen by proteases into peptides, and these peptides are presented on the surface of the cell. So this is why they are called antigen-presenting cell. And if a cell finds a pathogen, it does all these things, and then it moves from the space where it found the pathogen, usually it's somewhere in our body, to the lymph node, and there it presents these antigens, these co-stimulatory molecules, and these cytokines to T cells. And you have to imagine that there are millions of different T cells in our body all the time, and we need to find that one specific T cell that is able to fight that blue bacterium, and the specificity is generated by the T cell receptor. That is this green and gray structure. And if the T cell receptor matches to that blue bacterium, it is able to bind to the peptides from the pathogen. That's the blue dot in the middle. And these are presented on so-called MHC molecules, major histocompatibility complex proteins. And if this happens, this just means some activating signal gets transduced into the nucleus of the T cell so that it can become activated. And the co-stimulatory molecules lead to both cells even sticking closer together, sticking longer together, and also the activation of the corresponding molecule that binds to the co-stimulatory molecule, that is the blue one, leads to the transmission of an even stronger activating signal into the nucleus of the T cell. And this results then in the activation of the T cell. This is what we call this three-signal theory of T cell activation. So the first signal is the presentation of peptides from the pathogen via these MHC molecules to the T cell receptor. The second signal is the co-stimulatory molecules, which, as I've said, is just a summarization of what I tell you, what I show you here is something that I have deemed foreign and dangerous. And the cytokines then tell the cell what to do, yeah? So the first two signals just tells the cell, the T cell, signal 1 means, okay, my T cell receptor is of use to fight this, whatever it is. And the signal 2 means, okay, the dendritic cell or the antigen-presenting cell has decided I should make an immune response against that structure. And then the T cell doesn't know, is it a bacterium, is it a virus, is it a parasite? And this is the information that gets transmitted via the cytokines. And then they differentiate into different types of T cells, which we will look at in the second lecture. And then they can efficiently fight that pathogen. Yes, Alex.
You said that the signal 2 is like activating or helps the activation of the T cell. But are there also inhibitory signals?
Yes. You need to make an immune response to certain pathogens to fight them. But as I always says in my lecture, even the best immune response has to come to a stop at the moment, or you will start attacking yourself and become autoimmune once you have cleared all the pathogens. And therefore, we also have co-inhibitory molecules that are expressed, for example, by regulatory T cells, which we will learn in the next lecture, that can then be used to shut down immune responses towards certain antigens. There was the word antigen. Okay?
Thank you.
So here we have the three signals that are presented by the antigen-presenting cell, and only if all three signals are there, the T cell can be effectively activated. And a normal cell, fascinatingly, also shows antigens via different molecules. You see they look a bit different here. The MHC class 1 molecule looks different than the MHC class 2 molecule here, but they do not show signal 2, so you see there's no co-stimulatory molecule, and they usually do not produce any cytokines. So therefore there's a suboptimal stimulation, meaning that the T cell can only get activated by such a non-antigen presenting cell once it has been authorized to mount an immune response. So there's no initial activation of T cells, if you want to call it like this, by normal body cells, because these cells here are specialized in sorting out who is good, who is bad, and starting the immune response. Once it's going, it's a different matter, which we will see in the next lecture. So if the antigen-presenting cells recognize a pathogen like this virus, it has to start an immune response, meaning it has to recruit, so get other immune cells to the place of infection and activate them, meaning telling them what to do. And this is usually done, at least in part, by molecules which have a function in cell-to-cell communication in the immune system. And here we distinguish three classes. The first are cytokines, which we already talked about. These are small soluble proteins that communicate between leukocytes, so immune cells and non-immune cells. And examples are, for example, IL-1 beta and IL-6. IL is an abbreviation for interleukin. So this is a molecule that transmits information between meaning inter, and leukin meaning between leukocytes, between white blood cells. So molecules that transmit information between immune cells. And then we have, for example, TNF. This is an abbreviation for tumor necrosis factor. This is also a cytokine that was just named differently because it was discovered in the context of cancer and anti-cancer responses. Then we have interferons. Interferons are a subgroup of cytokines. So every interferon is a cytokine, but not every cytokine is an interferon. And interferons are small, fascinating molecules that activate a lot of function in these cells, but let's say their main function is to directly interfere with viral replication, so fighting viral infections. And they are called interferon alpha, beta, or gamma. Then we have chemokines. Chemokines are molecules that regulate the development and migration of cells in the context of an infection, and they can lead to differentiation of cells, but the main function is they get produced in large amounts at the place of infection, and the further away you are from the place of infection, the lower the chemokine amounts are, the concentrations are, and the immune cells have receptors for the chemokines, and they migrate towards the concentration of highest chemokine amounts, and then they find the place of infection. And on the way there, they also get activated and they differentiate to be ready to fight the pathogen once they reach the infection. And Hannah has a question.
Yes. So do I understand correctly that the antigen-presenting cells find the pathogens, for example, somewhere near our lung?
Yes, they are patrolling in our body to find pathogens.
Then they move into the lymph node. There they activate the T cells. And the T cells know where the infection originally was due to the chemokines.
Yes.
And then they travel back to the point where the antigen-presenting cell originally found the...
Yeah. So the cells, as I just said, most of the immune cells are found in the blood and also in the lymph system. And this is like a circulatory system in your body. And a T or a B cell needs 24 hours to pass every point in your body once. And then once they recognize some cytokines or chemokines, they know, oh, something is maybe wrong in that tissue here in Stefan's arm. And then they move out of the bloodstream into the tissue, and then they try to find the place of infection. And not all cells move, also certain cell types stay and produce more cytokines and chemokines. So therefore it's possible, because the body is very, very big for such a small immune cell. Okay. And chemokines, sorry, it is confusing, have different names. One that is easy is the MCP1, mast cell chemotactic protein 1. So a protein that recruits mast cells, but for example, interleukin 8 is also a chemokine, which was wrongly named as an interleukin, so a cytokine. So it's complicated and also confuses a lot of my students. So cytokines can act on cells by binding to specific receptors. So only a cell that has a receptor for a certain cytokine is able to register the information that there is a cytokine, and they can act in two different ways. The first one is autocrine. So here the same cell that initially produces the cytokine also has the receptor, and the cytokine binds to the same cell and regulates the cell in some kind of autocrine loop. Then we have paracrine, meaning that the cytokine binds to a receptor on a cell that did not originally produce that cytokine. This can be another dendritic cell, can also be a T cell, a B cell, any kind of cell. So it's an important distinction. Autocrine binding to the same cell that produced the cytokine, paracrine it binds to a different cell. Yes, Alex.
Do you have an example why a cell should produce cytokines which then bind to the same cell?
Yeah.
When is this beneficial?
Yeah, for example, I can tell you two off the top of my head. One is interleukin 10, so it's an anti-inflammatory molecule that reduces inflammation. This can tone down the activity of certain cells, also at later stages of infection when they're not actually needed anymore. And the other one is interleukin 2. It's an important survival factor for T cells. And once the T cells get activated, they have to differentiate, they have to proliferate, they have to form many daughter cells. And this is an exciting and also exhausting process. And then they push themselves by producing, for example, this interleukin 2 and helping themselves to survive a little bit better in this critical phase of the immune response.
Okay, thank you.
So it is complicated with these cytokines, and I just put one example here. We have the dendritic cell or the antigen-presenting cell in the middle, and it can produce different cytokines that lead to the formation of different T cells that then fight different pathogens. And here you can see that it is a combination of different cytokines that lead to the differentiation of different cells that then fight these different pathogens. And we also not only have pro-inflammatory cytokines, so that cytokines that lead to the induction of inflammation and an immune response, but we also have anti-inflammatory cytokines, which lead to a suppression of immune response, to a resolution of the immune response, and to wound healing. And this is complicated. This is just an example to show you how complicated it is, and I can tell you this is the most basic example. It can fill whole posters in, for example, our offices where we look at the interaction between different cells and what cytokine leads to what kind of response in what context. So it is, as always, very complicated. So chemokines, just one slide to chemokines. They can be grouped into three families. Just that you have heard this. They are called CXC chemokines, CC chemokines, and C chemokines. And this is just referring to their structure. So they have their proteins consisting of amino acids, and they have cysteines as part of their amino acid sequence. And these cysteines can form so within one molecule located cysteine bonds. And these CXC chemokines have two intramolecular disulfide bonds that are formed by two cysteines that are placed next to each other, but there is one other amino acid in between, which is designated as an X. Then we have the CC chemokines, where it's the same, but the one amino acid in the middle is not there. And then we have C chemokines that just have not that many residues and just form one disulfide bond. Structurally, these chemokines bind to chemokine receptors, and these are these typical 7 transmembrane area containing proteins. So they go like a snake 7 times through the membrane, and with their outside end they bind to the respective chemokine, and their inside end is coupled to certain proteins. These are G-coupled proteins that once the chemokine binds to the outside, there is a change in structure of the receptor, and then certain residues get exposed that can be phosphorylated, can get activated, and then these signaling cascades start that either lead to migration of the cell, to activation of the cell, or to differentiation of said cell. If you put this into a bigger context, once the antigen-presenting cell finds a pathogen, recognizes it as dangerous, and activates a T cell, this will start a whole type of a whole wave of immune responses, and the most important ones, at least for the long-term protection against said pathogen, is the activation of the T cell. These differentiate then into effector cells, and these in turn activate naive B cells to become antibody-producing plasma cells. But how this works will be the topic of the next few lectures. So I would like to come back to this slide again, because now we understand how the immune system differentiates between normal components of our body and the dangerous ones, meaning the ones that activate the pattern recognition receptors. And also, these epithelial cells are some kind of Frankenstein cells, again, between a barrier type cell that just prevents the pathogens from coming inside our body, but they also have like characteristics of immune cells, because they also, for example, express toll-like receptor 5 and can recognize flagellin, which leads to the activation of a signal or transmission of a signal into the nucleus of said cell. This drives then the expression of certain molecules, certain genes. Among them are the genes for some highly pro-inflammatory, meaning strongly fever-inducing and immune-activating molecules called interleukin-1 beta and interleukin-18. And since they are so strongly fever-inducing, some picograms, some nanograms of these molecules in your blood will leading you having 41 degrees Celsius of fever, and even dying from this. They have to be highly regulated, and therefore they are modulated or regulated by a system that is called the inflammasome. So the first step is, as I said, the recognition of the pathogen by the pattern recognition receptor, resulting in the transcription of the genes and the production of these molecules, IL-1 beta and IL-18, but they are not initially produced in their active form. They are produced as so-called proforms. And if you imagine this is the active molecule, then we have a small peptide that is attached to it, and this just prevents binding of the receptor to said molecule. So they are biologically inactive at this point. Once the cell now recognizes also flagellin on the inside of the cell, because this means that the cell has been infected by one of these bacteria. This binds to another sensor, which is called the NLRC4 NIP56 receptor. This is a NOD-like receptor, NLR, and this binds flagellin once it is inside the cytosol, and then these receptors aggregate and they form like a wheel-like structure that is called the inflammasome, that then results in the activation of caspases, so proteins that cleave other proteins, and these caspases then remove the small signal peptides from the proforms of IL-1 beta and IL-18, and then these are biologically active and can induce an immune response. They are secreted outside the cell, and then they can start an immune response. And because these molecules are so strongly activating the immune system, they are regulated by this two-step process involving the inflammasome. Yes, Alex?
Do only epithelial cells have the inflammasome?
No, not only epithelial cells have the inflammasome. They are like the first cells to come into contact with most of the pathogens. That's why they have it, to make the initial distinction between dangerous and not dangerous. But we find them, for example, in other cells of the innate immune system. So we find them in dendritic cells, we find them in macrophages, where it is again used to make the distinction between self and non-dangerous and non-self and potentially dangerous.
Okay, thank you.
So besides the NLRC4 inflammasome, that is how it's called, that recognizes flagellin, but also salmonella, which are flagellated bacteria, but also a type 3 secretion system. This is a system that injects certain proteins like a syringe into immune cells to inhibit their function. We have other types of inflammasomes. For example, we have the NLRP3 inflammasome. This uses a different adapter molecule, not NLRC4, but NLRP3, and it is a very promiscuous sensor that recognizes different pathogen-associated molecular patterns, so structures only existing in pathogens, but also damage-associated molecular patterns. So these are only produced or released from cells once they are stressed or damaged, and they also form the inflammasome or trigger the formation of the inflammasome and then the cleavage of those molecules. Then we have the AIM2 inflammasome, triggered, as far as we know, by Francisella novicide, a bacterium, and the MCMV virus, which causes infections that can be really dangerous in certain individuals. They use an AIM2 sensor that recognizes double-stranded DNA from these pathogens. And we have also the NLRP1 inflammasome that recognizes Bacillus anthracis, a pathogen that is really dangerous and also used for bioterroristic purposes, and also the lethal toxin from Bacillus anthracis. And it's not completely known how it works. We know that NLRP1 is involved somehow in the recognition, but if there's, for example, another pro-caspase 1,5 molecule involved and how it interacts, it's not so clear at the moment. So this is just to show you that there are different molecules, different types of inflammasomes, different sensors that allow these cells to differentiate between dangerous and not dangerous. So once we have activated these cells and we want to start an immune response, we have a problem, because usually the infection is in the tissue, like our bacterium here, but the immune cells are in the blood vessels or in the lymph vessels, and the vessels, the blood vessels, are covered also by epithelial cell-like cells. They are called endothelial cells in this case, and they also stick together very, very tightly, and they usually do not allow these cells to just cross the endothelium. So they have to somehow squeeze through there, and this works once the cells get activated by cytokines, and this results in changes in the morphology, so how they look, these endothelial cells, in their adhesive properties, so what can bind to them, and their permeability, so what they let through. And this results then in changes that are depicted by these molecules on the surface, and also the cells, as we have discussed before, the immune cells react to chemokines that are produced at the site of infection by changing their morphology, expressing certain ligands or receptors that allow the cell then to squeeze through the endothelium. And since this is a monocyte, it has to differentiate in the tissue towards a macrophage that can then kill the pathogen. And this is just a total overview, and this is actually a little bit more complicated, and I want to go a little bit more in detail as we have now understood it. So here we have again the endothelium, and you can see that this is an activated endothelium because there are chemokines, so these are the orange dots around, and this endothelium has already been activated because it expresses certain ligands that allow cells to bind. For example, a LFA1 can bind to a ICAM1 molecule. These are all adhesion molecules. This means these are molecules that allow cells to adhere together, to stick together, if you want to put it simply. And what first happens is once a cell finds these adhesion molecules and binds to them with the respective receptors, the cell starts rolling along the endothelium and it starts looking if it finds more of these molecules or if it also finds chemokines. And once it does, then these molecules can bind to each other. So the adhesion molecules can bind to the respective receptors. The cytokines or the chemokines can bind to respective receptors, and this then changes again the activation status of the cell. This results in signaling inside the cell, and the signal is, look, here it looks like on the other side is an infection. You should try to cross that endothelium, and then we have a very firm, very strong adhesion to this place, and then the cell starts to migrate through the endothelium. You can see the cell squeezing through. This is what we call transmigration, and then it finds itself in a tissue with a lot of chemokines that bind to respective receptors, and as I told you, the chemokines also induce the differentiation of monocytes into macrophages that can then kill the respective pathogen. So now we have our macrophage, for example, in our tissue that is infected by a bacterium, and now it has crossed the endothelium and it starts to look for the pathogen, and it does so by binding certain receptors. This can be, for example, complement receptors. Then the cell starts to flow around said pathogen until it's completely enclosed, and then the pathogen finds itself in the dark inside the macrophage, and then it is in a vesicle that we call a endosome. And this endosome then fuses with lysosomes. These are these dark blue circles that contain a lot of proteases, so proteins that degrade other proteins, and this then results in the destruction of that pathogen, and it is then degraded into peptides. So it gets destroyed. So we have gotten rid of this one bacterium, but there may be more, yeah? There may be a whole colony of them, and we need to get rid of all of them if we want to fight that infection. And therefore, the cell, once it has been activated by the pathogen, it starts to produce cytokines and chemokines. And this results in the activation of the immune system, and the activation of the immune system is accompanied by a process that we call inflammation. And this is what we want to look at again now. And what I told you is not only true for macrophages, but for example also for neutrophils and also for dendritic cells. So inflammation is something that has been known for over 2000 years by now. And it was discovered by characterizing the 5 cardinal signs of inflammation. So here we have our little scientist, and he has an infection with a bacterium in his finger, and therefore in his finger locally, so only in his finger, because only there the bacteria are at the moment. The processes I just told you about take place. The macrophages get activated, they produce these cytokines and chemokines and induce an immune response, and this leads to inflammation. And this inflammation has 5 signs. So first one is heat. So you will feel hot. You notice once you are sick, you feel hot. Then the infected tissue can turn red. You also notice. It doesn't have to be the whole hand, but can be only the one finger or just a few centimeters. Then it will start to hurt, and it will swell. So it will get bigger. The tissue will increase in size. And the last one is called loss of function. So meaning if your hand is inflamed for longer times, you cannot use that hand anymore. So that's why he has this bandage. Yeah. So these are the 5 signs of inflammation: heat, redness, pain, swelling, and loss of function. And since they were discovered in Rome, in Latin, they are called calor, rubor, dolor, tumor, and functio laesae. The Alex has a question, I think.
Yeah, you said that this little scientist is sweating because it's getting hot, or there is heat involving, but there's also local heat, or because you have a high flow of blood, for example.
Yeah, first it starts with a local infection, and he will only feel heat in his finger, but later on, if it gets more severe, I just did this to make this more clear.
Yeah.
Okay, so the first 4 signs of inflammation were discovered by Celce. He was, I always mix it up, I'm sorry if I mix it up now. He was a Roman doctor, encyclopedist that discovered a lot of things and wrote a lot of books at that time. And the 5th sign of inflammation was discovered 120, 150 years later by Galen. He was a Greek doctor that worked in Rome. That's why I always mix them up. And he observed that if inflammation takes place for longer time, it results in loss of function. So how do these 5 signs of inflammation now come about? So here we have again our tissue that is infected with, for example, a blue bacterium, and this gets recognized now by the macrophages. They get activated and they produce cytokines and chemokines. And I think Hannah had a question here at this point.
Yes. So before that, you always showed us a dendritic cell as an antigen-presenting cell, and now you are showing us macrophages. So the question that I have is, can these 2 be used interchangeably? So are there also antigen-presenting cells, like dendritic cells, in here? And do these macrophages also present the information then to the other cells? Because here you only show the cytokines and chemokine secretion.
That is a good question. Yes, both dendritic cells and macrophages are so-called professional antigen-presenting cells because they take up these pathogens not only to degrade them, but also to show the peptides on the MHC molecules. They can be used mostly interchangeably. The dendritic cells are better at initial T-cell activation. The macrophages are a bit better at taking up pathogens and destroying them. They can only to a certain extent activate T-cells that have never been activated, but they are, for example, very good at reactivating T-cells. So that there's a bit of differentiation. But for your purposes at this level, they can be used more or less interchangeably. Okay. So once this happens, these cytokines and chemokines produced by the activated macrophages then act on the endothelium of the blood vessel. The blood vessel gets bigger and more permeable, and a lot of stuff and a lot of fluid comes out. So this results in heat because of better or more blood flow. And then, as we just learned, the immune cells start migrating into the tissue, and this leads to a swelling because now we have more mass in the tissue than there was before. All these cells and all the fluid have to go somewhere, and this leads to then the swelling and the pain and the redness is, as I said, from the increased flow of blood. And if this then holds on for longer periods of time, we will lose the function of said tissue or organ. So here we have a clinical example of inflammation that is quite, I think, good to understand. And I asked the 2, do you see the inflammation? I think it's quite obvious. Yeah. You see the inflammation.
We have a swelling, we have a redness.
Yeah.
Probably pain.
Mm-hmm.
Yeah.
And also loss of function, or because if you imagine you want to swallow something, that could be a problem, yeah. That was also scared the doctors when we were at the hospital with that child. It's actually my oldest son, and he has a pteroglossal cyst. So this is something that is not that uncommon. So it happens in your individual development, that you go through all the developmental stage. So at one point you look like a small frog, and you also have some gills that are at least initially installed in your body, and then they get taken down. And in some children this doesn't work, and then we have some cysts that remain there. And these cysts are places where that are not correctly or not very well connected to the rest of the body, to the immune system, and therefore there we have a lot of colonization with pathogens, and this leads to frequent infection, and then the tissue gets inflamed. And you can see it is red. You can see, you could feel that it is warm, it is swollen. That's the tumor. He said it didn't hurt, but I don't believe him, because he didn't want to go to the hospital at that point, and it will lead to a loss of function. And that was actually what scared the doctors the most, because it's so close to the arteries in the throat. That what makes it difficult. Yeah, this usually gets then treated with antibiotics to help fight the infection, and then once it's not inflamed, this cyst has to be removed in a surgical procedure. If the immune system, or if the innate immune system does not work correctly, we have certain diseases that we will also look in a future lecture. But just as a teaser, here you have, for example, a patient with granuloma. So he or she has a defect in the innate immune system, which results in these cells being not able to efficiently fight some pathogens. And what the immune system does, it starts fighting the pathogens, but it cannot get rid of them because it has a defect in some mechanism. And then at some point, the immune system gives up and it encapsulates the pathogens, and therefore you have these pimple-like structures under the skin. And these are big aggregates filled with pathogens that are engulfed by immune cells, and is a last resort measure that just prevents the pathogens from spreading more. They're just confined in these granuloma. And here we have another example. This is a infection in the throat with Candida albicans. This is a fungus that everybody with a normal immune system doesn't have problems with, but certain immunodeficient patients have. And in small children, such an oral infection with Candida albicans, which is called thrush, is the sign of immunodeficiency, which could be inherited or developed. And in grown-up patients, usually it's men in their late 20s or in their early 30s that come to the clinic with this. This is usually a sign of an infection with HI virus. So these patients are then immunosuppressed, immunocompromised, and cannot fight these fungi anymore. So we have to look at a few more cells, but we will do this very quickly. So we have neutrophils. As we learned, these are the most frequent cells in the human body. They can phagocytose, so take up pathogens. They can produce reactive oxygen species. So this is a collection of molecules that contain oxygen atoms, and some of them have like single isolated electrons, and electrons never want to be single. They always want to be in pairs, and they are very selfish. If they see another pair, they rip just one electron out to be a pair again, but this leaves the molecule where the other electron was ripped out with one electron less, and by this damaged, and this is then produced in large amounts. These molecules are produced in large amounts to damage, for example, pathogens in early immune responses. Then they can also degranulate, so they can release the content of their granula, and these contain like hydrolytic enzymes, so proteins that degrade other proteins. Myeloperoxidase is a molecule that produces more reactive oxygen species. Lysozyme is a molecule that degrades certain pathogen structures. Lactoferrin is a molecule that removes iron molecules, which are important for the enzymatic activity of certain bacterial proteins. And alkaline phosphatase is a molecule that is also important for the activation and migration of these neutrophils, but its function is not completely understood. And what is very cool and only known for certain years is that these neutrophils can degrade their nucleus, and shoot their DNA like Spider-Man or like spiders as a net and trap certain pathogens. And this is here shown again. Here the yellow cell on the left upper hand is a neutrophil, and the green one is the DNA, and the purple one are bacteria that are then trapped in this DNA, and the orange cell on the right side is a bystander erythrocytes or red blood cell. And these nets, neutrophil extracellular traps, are consisting of elastase, which keeps the whole mixture elastic, cathepsin G, which degrades like the outer shell of certain pathogens, and myeloperoxidase, which uses hydrogen peroxide to generate more reactive oxygen species like these chloride anions there. And this is why we also have so many of them, because once they start shooting their DNA around, of course they die, and therefore we have so many of them. They are like disposable items in fighting these pathogens in a way. So the last set of cells we want to look at are mast cells, eosinophils, and basophils. You can see they all have a lot of these granula inside. That's why they are called granulocytes, and they can be stained because the content of these granulas is slightly different. And I didn't just make them these colors because I found it nice to look at. These are the colors these cells stain with certain dyes in tissues. So the eosinophils will be red, the basophils will be orange, and so on. And they need different factors like growth factors and cytokines for the differentiation. You can see they are quite similar. They all need IL-3. Then the mast cells need stem cell factors IL-4 and IL-9. The eosinophils need IL-5 and GM-CSF, and the basophils need IL-3, IL-5, and GM-CSF. They all have similar function. We will look in the next slide at, but what differentiates them is their anatomical location. So the mast cells are by far the most frequent of the three. We find them in the surfaces or the contact areas in all our body where we have potential contact with the outside, like under the skin, in the lung, and so on. They can directly fight pathogens there. The basophils we only find in very low numbers in circulation if they are not needed, and the eosinophils we find in the respiratory tract, in the gut, in the urogenital epithelium. But once they get activated, once they are needed, then they can proliferate, and we can find much higher numbers of these cells. And that's why they also, especially the eosinophils and the basophils, are used in clinical diagnosis to find patients that have, for example, parasite infections or allergies. These have usually increased numbers of these cells. And you can also see that their nuclei look a bit funky. That's why they're also called polymorph nuclear cells, because they have like lobed structures in the nuclei. So what do? They are specialized in fighting multicellular parasites. And as we learned in the beginning, these multicellular parasites are very, very big. So the smallest ones are some millimeters, and the biggest ones are 12 meters. And these are structures that are way too big to be taken up by a single cell. That's why, for example, the mast cells, but also the eosinophils and the basophils recognize these parasites or their surface with certain adapter molecules that we call antibodies. And then they release the contents of their granula, which is, as we learned, a cocktail of toxic molecules like proteases that degrade these parasites or their toxins. We have heparin, which is a coagulation inhibitor, also a blood clotting inhibitor. We have serotonin, which is a mediator in the nervous system that triggers, for example, muscle contractions that then expel the parasites from the gut. We have histamine, which is an inflammatory molecule, and they also produce a lot of cytokines to attract different immune cells. And this happens very quickly. So if you activate these mast cells, they can degranulate within seconds. But what also happens is that there is a second wave of molecules that is produced. So the first wave I just told you about is always ready. So like a mast cell is like a grenade. If you pull the pin, it will just explode with all these preformed molecules. And then we have a collection of newly generated, newly secreted molecules after certain minutes or hours. And these are usually molecules that are derived from lipids like prostaglandins, thromboxanes, leukotrienes, platelet-activated factors. And these are molecules that induce very strong inflammation over very short distances in our body. So this leads to a very strong immune response that usually recruits a lot of immune cells, and then the immune system is even able to get rid of these very big parasites. If these cells get activated not by parasites, but by structures, proteins from our surrounding that are actually not dangerous, this results in allergies. And I've shown you some examples here. You have like eczema. This is an allergic inflammation triggered in the skin by contact with these allergens. We have allergic conjunctivitis, so an inflammation of the eye. We have a food allergy, like this person drinking milk and feeling sick to the stomach. And here you have a bee sting on the right side, where the whole foot is swollen after being stung by a bee. So this is a big problem since this is increasing in frequency in our society. So if we come to the summary, what I showed you today is that our immune system consists of different barriers. We have the anatomical barriers, the complement system, and the antimicrobial peptides. We have the innate immune system, and we have the adaptive system, immune system, that we have not started to look at. And what all these barriers do is they, one by one, reduce the amount of pathogens that are able to invade the sterile inside of our body. And the first three barriers we talked about today are able to eliminate like 95-98% of all pathogens, which is very good, but not enough, because the last 2 to 5% where we need the adaptive immune system for would definitely kill us. And this also happens in patients that do not have an adaptive immune system. And how this works, we will look in the next lectures. So if we summarize the principles of the innate immune system, we know that it is immediately available. It can react within minutes or hours. And so these cells are, what I mean with immediately available, they are there in all of us. They have the same set of receptors in all of us, and they can react very quickly. But they rely on recognizing pathogens using only a limited number of pattern recognition receptors. So there are maybe 3 dozens of these receptors. They are okay at fighting and recognizing and fighting pathogens, but they're not perfect. And with one exception, which we'll look at in one of the later lectures, they do not form memory responses. So you can, if you get contact with a pathogen again, a second or a third time, you will form the same, more or less the same innate immune response 2 or 3 times. And this is not very effective, as we will learn. The adaptive immune system has different characteristics, and we will start looking at this in the next lecture. With this, I'm at the end. I will ask the 2 of them if they still have any questions. No? Everything was discussed.
No, thank you.
Okay, then I hope you 2 learned a little bit. It's not so much probably because you know this already. But I hope that you listeners out there learned a little bit about the immune system, and we thank you for watching, and I hope to see you in the next video where we'll start looking at the T cells. With this, stay healthy, stay safe, and stay hungry for knowledge. See you.
See you. Bye.
Welcome to the main gig. 1 hour later. That was quite charming, huh?
Yeah, I would say this was really charming.
I can tell you, mice can be treated very well against a lot of different diseases.
Yeah.
They will never develop. Meanwhile, yeah, there are thousands of drugs that work perfectly in mice.
Yeah, but maybe not in humans because they are different from our. Und Galen, griechischer Arzt und Naturforscher.
Ja, richtig. Correct. I know a little bit of stuff. A little bit, yeah. Thank you, Alex. Okay. Then bind. PowerPoint hates me today. PowerPoint hates me. Schlabberhose.
Jeder hat eine Schlabberhose
Did you see it,
an.
Alex?
Schlabberhose.
Schlabberhose, we saw
Jeder
it.
sollte Schlabberhose tragen.
Yes. Welcome to Easy Immunology. My name is Hannah.
And my name is Alex. And
And my
today
name
we
is-
are listening to Stefan.
Das war so nicht abgesprochen. That was not what we planned.
Okay, then just again.
Okay, then just try.
Just again.
Again, sorry.
Welcome to Easy Immunology. My name is Hannah.
My name is Alex.
And now we don't say anything anymore.
No, I just waited for you.
Please announce your plans in advance, Alex.
Okay, I will just say my name. I know this is hard enough.
And left. Yeah, no, right. No, wait. Sorry.
Another outtake.
I can start with, hi, I'm Hannah, a first-year PhD student at Stefan's lab. And then Alex can say, I'm also a PhD student at Stefan's lab. And then Stefan can say, I'm the famous Stefan that everyone has talked about before.
I like this as an outtake.
Biologists. I wanted to say biologists. Yeah. Okay, again.
But you didn't.
I know. It's difficult. Words coming out of
I
my
know.
mouth in a different language is even more difficult.
Hi, welcome to Easy Immunology. I'm Hannah, who nobody knows. But then
Yeah.
there's Alex and Stefan.
Let's try again. Alex, what are you saying? Just your name or?
Just my name.
Okay.